Today we're going to talk about chapter 12, which is on gene expression, and we will jump right into what we actually mean by gene expression. Let's start with a quote. Discipline is choosing between what you want now and what you want more. Gene expression is another word for making a protein. We're going to go through two different steps of a process to make that protein. So when I'm talking about whether genes are expressed, I just mean whether they're turned on. And we turn them on by going through transcription and translation. If we take a look at the caterpillar and the butterfly, I want you to be reminded that this caterpillar becomes that butterfly. The caterpillar has all of the same DNA here as it does here. So what happens inside this cocoon? What happens is that the caterpillar genes, if you will, get turned off and the butterfly genes get turned on. That is the difference in gene expression between two different stages of this organism's life. And we often call that differential gene expression. Just means of the thousands of genes that this organism has, certain ones will be turned on here and others will be kept off. Certain others will be turned on here and others will be turned off. But I do want you to think about what a cool process that is. What in the world is happening inside that cocoon that tells genes to turn on and off? Pretty amazing. Okay, so what are the steps to this and what does a gene look like? A gene is a piece of DNA. It's always a piece of DNA that is transcribed into RNA that will eventually make they say a functional product. And for the most part, that functional product is a protein. There are certain genes that don't go all the way to making a protein. They stop at making RNA. For instance, tRNA and rRNA, which we will see more of in following lectures. This idea of expressing a gene is summed up in what's called the central dogma. The central dogma is this, that DNA is used to make RNA. That RNA actually has to be processed, and that's what we see from this stage to that stage. And then the mRNA is read or translated and a protein is made. And as we said in the previous slide, sometimes we stop at the RNA stage, tRNA or rRNA. We don't move all the way to proteins. If you take a look inside the cell at where these things are happening, this is a bacterial cell. Let's just take a look at this one in particular. Here's the DNA, here is the RNA, and the RNA is then used to make a protein. So this little blue part here is a protein. It's called a polypeptide. That's just a small piece of a protein. So the process here of moving from DNA to RNA is called transcription. The process of moving from RNA to a protein is called translation. Gene expression involves transcription and translation. Making a protein, transcription, translation. When I turn a gene on, I am expressing it by going through transcription and translation. If we take a look over here at this cell, we'll see the same steps of transcription and translation, but a few changes are that the transcription part happens inside the nucleus. We have a nucleus, the bacterial cell does not. Once transcription occurs, the mRNA product is going to be modified in some way. You don't see that modification over here. It doesn't happen in bacterial cells. Once it's modified, that mRNA will now leave the confines of the nucleus and it'll move out into the cytoplasm. And that is where translation occurs. It hooks up with a ribosome, which is on the endoplasmic reticulum, not shown here. and again, the protein is made. So what we're going to see is there are a few more steps that happen in eukaryotic cells that we don't see happening in prokaryotic cells. If we want to take a look at the gene in more detail, right? The gene is always a piece of DNA. So this is all DNA. There are parts of it that help me to make a protein, that help me to make mRNA. But there are other regulatory regions as well. So this region right here is what we call a regulatory sequence. It might be later referred to as a switch, where transcription factors sit. And then we have something called a promoter. The promoter is this flag, the red flag that says, this is where I want you to sit down and begin transcribing. And so this is called the transcribed region. But I do want to point out, as you'll see later, that the promoter is where something called RNA polymerase sits. Okay, so we have the regulatory region, then the place where RNA polymerase sits. RNA polymerase is going to read through the transcribed region. And then there's another terminator sequence at the end. And this just signals to the transcription machinery that, hey, you've gotten to the end of a gene. It's time to stop. I will pick up in the next video with a more detailed description of transcription. Thank you.